perarakan masuk ke Dewan. Yang berbahagia, Academician Professor Emeritus Tan Sri Datuk Dr. Syed Jalaluddin Syed Salim, Pro-Chancellor Universiti Putra Malaysia, diiringi div-div kehormat. Para hadirin dimohon berdiri. Nyanyian lagu Negaraku dan Putra Gemilang. Para hadirin dipersilakan duduk. Mawar merah berjajar 5 di bawah sarang burung tempua. Salam sembah pembuka bicara, selamat datang untuk semua. Yang berbahagia, Academician Professor Emeritus Tan Sri Datuk Dr. Syed Jalaluddin Syed Salim, Pro-Chancellor Universiti Putra Malaysia. Yang berbahagia, Profesor Insinyur Dr. Hang Tuah Baharudin, Timbalan Naib Chancellor Jaringan Industri dan Masyarakat Universiti Putra Malaysia. Yang berbahagia Profesor Dr. Shuhaimi Mustafa, Dekan Fakulti Bioteknologi dan Sains Biomolekul. Yang kita raikan, yang berbahagia Profesor Dr. Foo Hooi Ling, Pegawai-pegawai kanan universiti dan ahli pengurusan fakulti, tetamu jemputan, wakil-wakil kementerian dan rakan-rakan industri, para pegawai akademik dan sidang hadirin yang dihormati sekalian. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Selamat datang diucapkan ke Majlis Syarahan Inaugural Profesor Dr. Foo Hooi Ling, The Myths and Fantasies of Lactic Acid Bacteria. What is next after probiotics? Bagi memohon keberkatan majlis pada pagi ini, dipersilakan Yang Berusaha Dr. Ahmad Muhaimin Roslan untuk memimpin bacaan doa.
Assalamualaikum warahmatullahi wabarakatuh. Al-Fatihah. A'udzubillahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Ar-Rahmanir Rahim. Maliki yaumiddin. Iyyaka na'budu wa iyyaka nasta'in. Ihdinas siratal mustaqim. Siratal ladzina an'amta 'alaihim ghairil maghdubi 'alaihim waladdallin. Amin. A'udzu billahi samil alim minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala asyrafil anbiya wal mursalin wa ala alihi wa sahbihi ajma'in. Ya Allah, Ya Rahman, Ya Rahim, Engkaulah yang mempunyai segala kepujian. Engkaulah yang berhak menerima segala kesyukuran. Engkaulah yang memiliki segala pemerintahan. Di tangan Engkaulah segala pengurusan, dan kepada Engkaulah kembali segala urusan. Kami memanjatkan kesyukuran kepada-Mu, Ya Allah, kerana mengizinkan kami untuk mengadakan dan berada di dalam Majlis Syarahan Inaugural yang berbahagia Profesor Dr. Foo Hooi Ling yang bertajuk The Myths and Fantasies of Lactic Acid Bacteria. What is next after Probiotic? Ya Allah, Rabbus Samawati wal Ard, bersempena dengan Majlis Syarahan Inaugural ini, kami memohon taufik dan inayah-Mu. Berikanlah kami kekuatan dan ketekunan dalam perjuangan mencari ilmu pengetahuan, menyebarkan informasi dan berkongsi hasil penyelidikan kami untuk memupuk komuniti masyarakat yang berpengetahuan luas dan berkemahiran tinggi dalam suasana kehidupan yang muhibah. Di samping itu, Ya Allah, bukakanlah hati kami, lapangkanlah dada kami untuk menerima ilmu pengetahuan serta bimbingan dalam membentuk kami menjadi ilmu insan yang berilmu dan berbakti. Ya Allah, yang Maha Kaya dan Maha Bijaksana, kami memohon kepada-Mu, Ya Allah, agar Engkau berkati majlis ini dengan limpahan pemurah dan penyayang-Mu, agar majlis ini berjalan dengan lancar dan baik. Semoga usaha kecil kami ini memberi faedah dan kebaikan kepada semua, dan kurniakanlah kejayaan dan keberkatan untuk mengiringi usaha murni kami ini. Allahumma inna na'udzubika minal barasi wal junun wal juzami wa min sayyi'il asqam. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azaban nar. Wa sallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi ajma'in. Walhamdulillahi rabbil alamin. Taqabbalallahu minna wa minkum.
Amin ya rabbal alamin. Majlis merakamkan ucapan terima kasih kepada Yang Berusaha Dr. Ahmad Muhaimin Roslan atas bacaan doa sebentar tadi. Hadirin sekalian, dengan segala hormatnya, majlis ingin mempersilakan Yang Hormat Profesor Datuk Dr. Roslan Sulaiman, Naib Chancellor Universiti Putra Malaysia, untuk mempengerusikan Majlis Syarahan Inaugural ini secara rakaman, dan seterusnya mempersilakan Yang Berbahagia Profesor Dr. Foo Hooi Ling untuk menyampaikan syarahan beliau. Sama-sama kita saksikan rakaman pembukaan beliau.
Terima kasih saudara pengacara majlis. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Yang berbahagia Profesor Dr. Shuhaimi Mustafa, Dekan Fakulti Bioteknologi dan Sains Biomolekul. Yang berbahagia Profesor Dr. Foo Hooi Ling yang akan menyampaikan syarahan beliau pada pagi ini. Pegawai-pegawai kanan Universiti Putra Malaysia, Ahli Pengurusan Fakulti Bioteknologi dan Sains Biomolekul, tetamu-tetamu jemputan, rakan industri, para staf dan pelajar, sidang penonton yang dikasihi sekalian. Alhamdulillah, syukur kehadrat Allah kerana dengan limpah kurnianya, kita masih lagi diberi peluang untuk bersama-sama bagi meneruskan usaha memartabatkan keserjanahan ilmu pada pagi yang barakah ini. Izinkan saya untuk membaca latar belakang Profesor Dr. Foo Hooi Ling dalam majlis yang gemilang ini. Dr. Foo Hooi Ling dilahirkan pada 7 Februari 1968 di Sungai Bakap, Pulau Pinang. Beliau merupakan anak sulung daripada 4 adik-beradik kepada pasangan mendiang Encik Fu Wing Tong dan Puan Koi Su Sim. Beliau telah berkahwin dengan pasangannya, Profesor Dr. Loh Teck Chuan, pengarah Pusat Transformasi Community University, UPM. Dan yang berbahagia dikurniakan dengan 3 cahaya mata perempuan, Loh Xiao Tian, Loh Xian Ting dan Loh Xiao Zuan. Prof. Dr. Fu mendapat pendidikan awal di Sekolah Jenis Kebangsaan Cina Xinhua, Parit Buntar, Perak. Seterusnya, beliau menyambung persekolahan di Sekolah Menengah Kebangsaan Methodist, Parit Buntar, Perak, sehingga ke peringkat SPM pada tahun 1986. Beliau kemudiannya meneruskan persekolahan ke peringkat STPM di Sekolah Menengah Kebangsaan Panglima Bukit Gantang, Parit Buntar pada tahun 1988. Usai zaman persekolahan, Dr. Fu meneruskan pengajian di Universiti Pertanian Malaysia dalam bidang biokimia dan mikrobiologi pada tahun 1989. Beliau memperolehi ijazah sarjana muda sains dengan kepujian pada tahun 1993 dan menyambung pengajian diploma pendidikan di Universiti Melaya pada 1993 dan telah menjadi seorang guru bertauliah pada tahun 1994. Beliau seterusnya dilantik sebagai tutor di Jabatan Bioteknologi Fakulti Sains Makanan dan Bioteknologi UPM pada tahun 1995 dan menerima beasiswa untuk meneruskan pengajian Doktor Falsafah di College, University of London yang kini dikenali sebagai Imperial College at United Kingdom dan memperolehi ijazah PhD pada tahun 1998. Kerjaya akademik Prof. Dr. Fu bermula pada tahun 1998 apabila dilantik sebagai pensyarah di Jabatan Bioteknologi Fakulti Sains Makanan dan Bioteknologi UPM. Beliau telah menerima kenaikan pangkat kepada Profesor Madya pada tahun 2004. Kini beliau merupakan Profesor di Jabatan Teknologi Bioproses Fakulti Bioteknologi dan Sains Biomolekul UPM bermula tahun 2017. Berbekalkan pengalaman lebih 23 tahun dalam pengajaran dan penyelidikan bioteknologi, kepakaran Prof. Dr. Fu telah digunakan untuk menyelia lebih daripada 52 pelajar siswazah dari pelbagai negara dan telah berjaya menggraduatkan 42 pelajar siswazah di mana 13 orang pelajar siswazah telah bergraduat dengan ijazah PhD. Secara tambahan, beliau juga telah berjaya memperolehi penajaan syarikat swasta berjumlah RM22,000 Malaysia untuk pelbagai hadiah anugerah pelajar cemerlang program bioteknologi. Dengan kerjasama rakan sekerja di UPM, Prof. Dr. Fu telah memohon dan memperolehi lebih daripada 18 geran penyelidikan dalam jangka masa 23 tahun perkhidmatan beliau di UPM. Secara kumulatif, beliau telah menerima lebih daripada RM5 juta Malaysia geran penyelidikan daripada UPM, Kementerian Sains, Teknologi dan Inovasi Malaysia, Kementerian Pendidikan Tinggi Malaysia dan juga Kementerian Pertanian Malaysia. Sebahagian daripada geran pendidikan telah digunakan untuk membangunkan dan melengkapkan 3 makmal penyelidikan UPM, iaitu makmal penyelidikan mikrobiologi, makmal bioteknologi mikrobial dan makmal bioteknologi industri. Pada tahun 2009, Prof. Dr. Fu telah berjaya memperolehi geran penyelidikan khas iaitu Special CRDF Bridging Fund berjumlah RM3.2 juta Malaysia daripada Malaysian Technology Development Corporation Sedia Berhad untuk mempertingkatkan pengeluaran inovasi penyelidikan yang telah dipatenkan. Sejumlah 1.7 juta ringgit Malaysia daripada dana CRDF telah digunakan untuk membangun satu loji perintis semi dan makmal penyelidikan yang dikenali sebagai Probiotic and Cancer Therapeutics di Institute Bioscience UPM. Pada tahun 2010, satu grant Technofund berjumlah 1 juta ringgit Malaysia telah diperolehi daripada Kementerian Pertanian Malaysia untuk membangunkan satu loji perintis untuk pengkomersialan produk inovasi di mana paten inovasi telah dilesenkan kepada sebuah syarikat swasta. Prof terlibat secara aktif dalam bidang penyelidikan bioteknologi industri, mikrobiologi gunaan, bioteknologi enzim dan makanan, bioterapeutik, teknologi biopemisahan dan penulinan. Justru beliau telah menerbitkan lebih daripada 300 kertas kerja dalam jurnal saintifik terkemuka dan prosiding persidangan kebangsaan serta antarabangsa. Beliau juga sering dijemput untuk menyampaikan syarahan di persidangan kebangsaan dan antarabangsa serta pembentangan ceramah industri. Selain aktiviti pengajaran dan penyelidikan, beliau juga terlibat secara aktif dalam aktiviti profesional. Sidang hadirin yang dimuliakan sekalian, dari segi penerbitan secara kumulatif, Prof. Dr. Foo telah berjaya menghasilkan 5 judul buku berkaitan dengan bakteria asid laktik dan lebih daripada 115 artikel jurnal untuk penemuan hasil penyelidikan. Dari segi penghasilan harta intelek, bersama dengan rakan sekerja UPM, Prof. Foo telah memfailkan sebanyak 33 paten di mana 20 paten difailkan sebagai ketua projek. Kini lebih daripada 12 paten dan satu cap dagangan ataupun trademark telah dikurniakan oleh Agensi Harta Intelek Malaysia, Australia, Singapura, China, Jepun, USA dan juga Korea. Beliau turut berjaya mencapai matlamat muktamad sebagai seorang penyelidik dengan mengkomersialkan 5 inovasi penyelidikan di mana 3 paten telah dilesenkan kepada syarikat swasta pada tahun 2005, 2010 dan 2016. Seterusnya, pada awal tahun 2022, 2 paten telah berjaya dijual terus kepada satu syarikat tempatan untuk mempromosikan industri bioteknologi di Malaysia. Merentasi lebih 23 tahun dalam kerjaya di UPM, Prof. Dr. Foo telah menerima pelbagai pengiktirafan dan anugerah, di mana 21 anugerah penyelidikan telah diterima daripada pertandingan penyelidikan kebangsaan dan juga antarabangsa. Antaranya ialah anugerah penyelidik cemerlang UPM dalam kategori pengkomersialan pada tahun 2006. Selain itu, beliau telah dilantik sebagai pemeriksa dalaman dan pemeriksa luar bagi tesis pelajar siswaza untuk pelbagai institusi pengajian tinggi di Malaysia. Berdasarkan pengalaman beliau yang luas dalam bidang bioteknologi, beliau turut dilantik sebagai ahli panel Board of Study bagi program Bachelor Science Bioteknologi yang ditawarkan oleh Infrastructure University Kuala Lumpur dari tahun 2016 hingga 2020. Prof. Foo juga telah dilantik sebagai Profesor Adjunct di Fakulti Pengajian Vocational Universitas Airlangga Indonesia untuk tempoh 2021 hingga 2022. Hadirin dan hadirat yang dihormati sekalian, Prof. Dr. Foo juga aktif dalam aktiviti pengembangan di mana beliau kerap dilantik sebagai penasihat dan konsultan bagi badan berkanun, badan bukan berkanun dan syarikat swasta. Beliau juga sering dilantik sebagai para panel teknikal bagi grant penyelidikan di peringkat fakulti, UPM, MOSTI, MOHE dan juga Netherlands Organisation for Scientific Research. Untuk penglibatan aktiviti saintifik peringkat antarabangsa, beliau telah dilantik sebagai editor bagi Journal of Biocatalysis and Agriculture Biotechnology, iaitu jurnal rasmi Persatuan International Society for Biocatalysis and Agriculture Biotechnology. Prof. Dr. Foo turut menonjol dalam aktiviti Persatuan Saintifik Antarabangsa, iaitu Asian Federation Society for Lactic Acid Bacteria, di mana beliau merupakan salah seorang wakil Malaysia yang telah dilantik sebagai ahli lembaga eksekutif persatuan sejak tahun 2009. Beliau seterusnya dilantik sebagai Timbalan Presiden untuk tempoh 2019 hingga 2023. Beliau juga turut aktif dalam persatuan saintifik kebangsaan seperti Malaysian Society for Microbiology dan Malaysian Society for Lactic Acid Bacteria. Prof. Dr. Foo terlibat dalam aktiviti persatuan sejak tahun 2000 di mana beliau telah memegang pelbagai jawatan sebagai Setiausaha Kehormat, Berdari Kehormat dan Timbalan Presiden. Seterusnya, beliau telah dilantik sebagai Presiden MSLAB pada tahun 2021 dan berjaya memenangi tawaran sebagai pengerusi penganjuran 12th Asian Conference on Lactic Acid Bacteria yang akan diadakan pada 10 hingga 13 Oktober tahun ini di Kuching, Sarawak. Prof. Dr. Foo turut aktif dalam aktiviti komuniti di mana beliau telah dilantik sebagai Setiausaha Kehormat bagi Persatuan Sedang Rhythmic Gymnastic Angels pada tahun 2010 hingga 2013. Beliau turut dilantik sebagai Bendahari Kehormat dan ahli jawatankuasa penganjur bagi International Rhythmic Gymnastic Tournament anjuran SRA Club dari tahun 2010 hingga 2014. Seterusnya, beliau diberi tanggungjawab sebagai pengurus pasukan Selangor Amateur of Gymnastic Association untuk Lilac International Rhythmic Gymnastic Competition yang telah diadakan di Sapporo, Jepun pada tahun 2012. Beliau turut terlibat sebagai ahli jawatankuasa 13 Rhythmic Gymnastic Asian Championship pada tahun 2014 yang telah dianjurkan oleh Malaysian Gymnastic Federation. Sidang hadirin yang dikasihi, kita tidak akan lihat Prof. Foo untuk melakukan gimnastik, tapi sekarang marilah kita mendengar syarahan inaugural oleh Profesor Dr. Foo Hooi Ling yang bertajuk The Myth and Fantasies of Lactic Acid Bacteria. What is next after probiotics? Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Okay, salam sejahtera and salam satu Malaysia kepada yang berbahagia Academician Emeritus Profesor Tan Sri Datuk Dr. Syed Jalaluddin Syed Salim, Pro-Chancellor Universiti Putra Malaysia dan para hadirin sekalian. Saya mohon menyampaikan syarahan inaugural saya dalam bahasa Inggeris. A very good morning and salam satu Malaysia to all my distinguished guests and also yang berbahagia Academician Emeritus Professor Profesor Tan Sri Datuk Syed Jalaluddin and Syed Salim, Pro-Chancellor of Universiti Putra Malaysia, and yang berbahagia Professor Insinyur Dr. B.T. Hang Tua Baharudin, the Deputy Vice Chancellor Industry and Community Relation, Universiti Putra Malaysia, yang berbahagia Profesor Dr. Suhaimi Mustafa, the Dean Faculty of Biotechnology and Biomolecular Sciences, Senior Officers, Universiti Putra Malaysia, Management Members, Faculty of Biotechnology and Biomolecular Sciences, Academic Officers, distinguished guests from other universities, ministries, research institutions, and industrial partners. Staff, fellow students, and other distinguished guests, ladies and gentlemen, it is my great pleasure that Universiti Putra Malaysia is given me the honor to deliver my inaugural lecture entitled The Myth and Fantasies of Lactic Acid Bacteria. What is next after probiotics? Can somebody help me to do? Okay, right. Thank you very much. Okay, when I mention lactic acid bacteria, I think most of you do not know what it is, right? But when I mention probiotic, I think everybody know very well, all right? So that's the reason why I coined the probiotic in my inaugural title, okay? Okay, I like to ask questions during my lecture time, including today as well. So the content, all right, I listed down a few questions here. So after the lectures, you should be able to answer those questions if you go through together with me. So the first question, you all should know what are the paradigm shifts of lactic acid bacteria research and development. And the second question, what are probiotic and also their challenges. The third point, what are the derivatives of probiotics nowadays that you can find in the market as well. And the fourth, should we consume probiotic, postbiotic, or paraprobiotic? And the last one, how versatile the LDB can be a biotransformation agent to degrade the polymers of biomass to produce bioactive compound. Okay, another question. Can lactic acid bacteria be a promising biotransformation agent? Tools or alternative to achieve the sustainable goal that proposed by UN. So what are the sustainable goals? Altogether, 17 sustainable goals has been proposed in year 2015 to be achieved by the year 2030. And it is a call for all the countries to foster development while protecting our environment, including human beings. When you mention lactic acid bacteria, it is always associated closely with probiotic. And it shows that in year 2021, 61.1 billion sales of probiotic has been recorded. And it was projected to be 91.1 billion in 2026. It's a huge jump. So why? What are reasons that have driven the rise of the projection of the probiotic market? There are few. The first one is the change of the consumer behavior, whereby consumers nowadays look for convenient foods and the disposable income also has been raised. They can go for more convenient but nutritive food product. And the second point is the awareness of health promoting foods. Nowadays, because of the COVID-19 pandemic for the past 2 years, I think all of us, we change our lifestyle no matter we like it or not. Yeah? I think we all try to get, if possible, try to improve our health through the food consumption. And that's the reason why the probiotic product has been, one of the major alternatives or selection to improve our health. The subsequent point is driven by malnutrition in those poor countries like Africa. And as well as the old age and also non-communicable diseases like obesity, and also we try to have the high nutrition food if possible. And another point is investment by most of the company nowadays, because due to the driven of the consumer, they want different food which is more convenient to take. And actually, the probiotic market has been increased a lot in Asia Pacific region because due to the requirement or consumption of the functional food. So, let food be the medicine and let medicine be the food. That is a slogan nowadays that most of us would try to practice it. Okay, lactic acid bacteria actually we consume for a long, long time ago. before the civilization of human being, or by knowing what is the role of lactic acid bacteria in our food preparation, food preservation, and food production. So that's the reason why we have been consumed for ages, but they do not give, most of them do not give the deleterious, the adverse effect to human being. And therefore, it is known as generally regarded as safe by the FDA, USA. And at the same time also, they have been granted qualified presumption of safety, QPS status, by the European Food Safety Authority, or EFSA. Generally, lactic acid bacteria is a group of bacteria, and all of them actually come from different genus. And it is a bit special compared to other group of bacteria or microorganisms. And here, I listed here, 17 genus has been included in lactic acid bacteria group. Okay? And you can see that these are closely related genuses, and Bifidobacterium and Propionibacterium, they are far away, but they still included in the lactic acid bacteria due to the metabolism's characteristic. And for those on top of here, they belong to Firmicutes, a class of bacilli, and also order of Lactobacillus. And for the Bifidobacterium, it is very special, and it belongs to the phylum of Actinobacteria. So in year 2020, a polyphasic approach has been adopted by a group of researchers known as Zhang et al. They try to reclassify the group or genus of Lactobacilli into different, or further 25 of the new genera has been proposed. So in this case, most of the product that has been used, Lactobacillus, now they have to rename them with a new taxonomy. And I think for the registration purposes also, I think that one is another issue for the registration body to understand and to adopt the new taxonomy system. And at the same time also, Lactobacillus and Leuconostoc, genus has been reclassified as shown here. So this is a new classification, and those in industry, I think you should know and try to rename your culture now, for registration purposes. Okay, so all of them, doesn't matter what cities that they come from, all of them, they are gram-positive bacteria. That means that they stain in purple blue color. All right. Normally, bacteria can be differentiated by gram staining, either in the pink color or purple color. So all lactic acid bacteria, regardless what genus it is, where they come from, all of them are gram-positive bacteria. And it's a good thing about LAB because they do not form the spore. In terms of safety, it's much more safer. But of course, it does not last long compared to those bacteria can form the spore. And normally, they're present in either cocci, round shape, like the bubble or marbles, and also either in the rod shape, like sausage. All right? And they are non-motile, not like E. coli. And they are acid tolerant because they produce acid by themselves. And therefore, based on the acid production, we can reclassify them or further classify them as a homofermentative lactic acid bacteria or heterofermentative lactic acid bacteria. If they produce only lactic acid from the carbohydrate fermentation, it is known as homofermentative. But if they produce also other organic acids such as acetic acid, then they are heterofermentative. Why they produce lactic acid? Some produce acetic acid as well. Okay. Hello, testing. All right. Okay. So the heterogeneous of the organic acid production is due to the metabolism pathway that harbor in the cell. So this slide shows the 3 metabolism pathway that normally you can find in lactic acid bacteria. If they are homofermentative, means that they will produce lactic acid through glycolytic pathway, all right, or through this pentophosphate pathway. I hope that you still remember the pathway, some of you. Okay, I think you know you don't like the pathway, I know that. But actually they are very important because they are the pathway or chemical pathway that produce the end product that which can be taped on for further functional study. Okay, and if they're heterogeneous or heterofermentative, means that most likely they will go through this phosphokinolase pathway by producing lactic acid at the same time also acetic acid to further ethanol. And these end products are very important in the functionality of lactic acid bacteria. Okay, I hope that you're not hungry yet, right? Yeah? Okay, so this slide shows some of the foodstuff that you can find lactic acid bacteria. So this slide shows that these are the common genera of lactic acid bacteria that you can find in the fermented food or food preparation. Okay, we have embarked on lactic acid bacteria isolation and screening since year 2000, or start actually 1999 we have begun the isolation and looking for a good candidate for further study. So we have done some, we have tried to select a few fermented food that we can find in Malaysia. Why we use fermented food? Why not from the gut? Why not from other animal origin? Because due to one issue, which is halal issue. If our culture comes from food, we can try to not to worry about the halal issue. That's the reason why we begin with the isolation from food sources. And we obtained more than 300 presumptive isolates for our beginning of the process. And this culture actually listed here are frequently used, or can we say that they are the major player in our research activity down the road for 23 years. Okay, so this slide shows you a few characteristics, method for the identification of lactic acid bacteria. Okay, this is quite common, gram staining, and also this carbohydrate fermentation profile or pattern. From here, we can tell that we can differentiate that into genus level. And at the same time, also, we also try to check their growth profile or growth activity under different pH condition, different pH temperature condition, as well as the effect of different grow media. So from there, we can tell, well, we can differentiate them further. And in my research group, we also check for the enzyme activity, such as proteolytic activity, cellulolytic activity, and hemicellulolytic activity. Okay, and this one, actually, these two parameters can tell, in terms of functionality, they are different. But when we try to use 16S rDNA, which is molecular tools, to differentiate the LB, we cannot differentiate the 6 strains or the strains that we use for our study. And we actually being queried whether the culture we use in our study come from different strain or different individual, or they come from the same clone. So we stuck there for some time. So what we have done, right? We also try to use polyphasic genotypic differentiation method. And in last decade, it is not easy to differentiate strain level. Nowadays, because of a new next generation sequencing approach, you can sequence the whole genome easily. And it's quite costly, still costly, but compared to last time, the NGS or next generation sequencing is not commonly available, and it is very costly. So it imposed a lot of challenges to us to differentiate the 6 strains that we use heavily for our study, because for the publication, they query that. So what we have done, with the help of Prof. Dato Raha, she is not with me, she is a molecular biologist. With the help, actually, we managed to differentiate the 6 bacteriocyanogenic LAB into different strains, but it's quite tedious. Okay, we based on the ITS1 region, which is the 16S, 23S intergenic spatial region of ribosome, and from the sequencing of the ITS1 region, we found that 4 base pair variation into some of the strain that we study. So based on that 4 base pair variation, we can differentiate our 6 strain into 4 subtypes. Yeah? But still, this one have not resolved. Are they same or are they different? And we also try to check the ITS2 region. It shows that they are highly conserved, no different. So what should we do? We try to engage another approach based on plasmid profiling and also random amplified polymerism DNA analysis, a few of them, by using different primer, only we manage to differentiate them into the 6 strain here. So based on plasmid profiling, you can differentiate into 3 subgroups again. But based on RAPD, you can differentiate them accordingly. So we had to combine several genotypic classification, then only we can tell they are different. They are not clone. They are different individual. Right, so what are the applications of LAB? When you mention application of LAB, you can't run away from the food. Okay, because it was started, yeah, with the food for the application. So beside that, they also now been developed to become nutraceutical and functional or health supplement product. At the same time, also now they've been used to replace antibiotic as growth promoter for fish and animal husbandry. And last one, biotransformation is a new paradigm that I would like to introduce to all of you. They are versatile tools to transform many, many biomass polymer. Okay, LAB application in food industry, why they've been used for so long, and they are still being used to process or to produce the food, because they can use to preserve the food, at the same time also improve the nutritional properties, at the same time also they can enhance the flavor, the texture, and also the aroma that you want. You can tailor make. And of course, this is quite important to upgrade, yeah, the food become more nutritive and more bioavailable for the nutrition factors. And the last one is being commercialized to become starter culture for for yogurt drinks production, cheese, and etc. Okay, and kimchi as well. Right. Okay, this slide show many tick, right? Start from probiotic, prebiotic, I think you know very well, am I right? How about paraprobiotic and postbiotic? Are they new to you? Right? So, for the probiotic, I think you know very well. Actually, nowadays, the probiotic also has been classified or has been introduced as a psychobiotic and immunobiotic based on the name. Actually, based on functionality of probiotic that discover, those probiotic that can have the mental effect, then they call it psychobiotic. And for those can induce immunomodulation activity, and it is classified as an immunobiotic. How about paraprobiotic and postbiotic? They are different preparation. I will tell you more later on. And basically, yeah, for the postbiotic, nowadays also known as cytobiotic, which derived from my research group. And it also known as an immunoprobiotic if they can modulate the immune system. And for these two preparations, actually they do not involve viable cell or cellless, in other words. But these probiotic, they must have the viable cell in order to have the function. And prebiotic basically is not a cell. All right? Okay, if it's based on the probiotic definition, before that, actually, the one that we are using now actually derived from the initial probiotic definition, which is proposed by Lilly et al. 1965. So actually, according to Lilly et al., it is a substance produced by protozoa. It's not bacteria. It's another group of microorganisms that which can support and then support the growth of other microorganisms. So basically, that is the initial discovery. Later on, Fuller tried to propose another one, which is more on the animal production or husbandry effect. And later on, another one also based on effect, yeah, of the probiotic as well. So the last one actually quite close to what we use, which is proposed in year 2000, in year 1998. Okay, so what are the... Okay, so this is the currently we use commonly or well-accepted probiotic definition. And this actually proposed by the WHO, WHO 2001, and also adopted by our Kementerian Kesihatan of Malaysia as well. Okay, so what it is, actually it is live microorganisms, the keyword live, okay? And then when administered in adequate amounts, must be adequate amounts, okay? And confer health benefits on the host. So actually this statement to me is very general. And actually can be a controversial, in terms of application, in terms of rules and regulation, monitoring. Right, okay. In order to be a good probiotic, they have to fulfill a few selection criteria, such as you must know the origin of the microorganisms. And the second point, must be able to survive from gastric transit, because our gastric is very acidic. Most of the microorganisms cannot survive under that condition. The third point is does not harbor antimicrobial resistant gene. And this is really a killing point to most of the probiotic that available in the market. Because doesn't matter we like it or not, most of the bacteria will harbor certain MR gene. But the question is whether this MR gene is transmittable or not. If not, then you can apply as a probiotic. Right? So for the following point is, they must be able to adhere the gut epithelial tissue. That is another big challenge as well. You imagine that I throw you in the ocean, and that ocean got so many candidates around. Will you be able to attach together? It's a bit difficult, right? This is what happened, right? And after attachment, after they can survive, what they should do? They still have to give some beneficial impact as listed here. For example, antagonistic activity against pathogens, and they have to be able to metabolize cholesterol, one of the example, or they can tolerate to the lactose, and also they can have the immunomodulation activity. So this example can be used to, show the probiotic impact. Right, so this slide just show you a few, probiotic that has been available in the commercial. Most of them, they come from lactobacillus or bifidobacterium nowadays. And of course, in the food industry, a bit different. They use different species and different genus also for food production, including Leuconostoc, etc. So here is a bit unusual, right? E. coli. E. coli also can be claimed as probiotic nowadays. So this is a bit special strain. It's been adopted as probiotic. And now, if you check on the Google, you can check Google and online, you can find out now yeast also claimed as a probiotic. So now question, what is probiotic? Is it necessarily come from Lactic bacteria, or can be any microorganism that have beneficial effect, that fulfill the criteria that I mentioned just now? Right? So based on the general, definition of this one, actually anything can be probiotic. Right. Okay, how about prebiotic? Always people ask, what is prebiotic? Is it a microorganism base or is it a bacteria base? No, it is not. It is a food base. So probiotic or prebiotic actually is non-digestible food or carbohydrate residues that you can get from the plant-based food. And but upon consuming that prebiotic, actually it gives impact to the beneficial bacteria to grow well in your gut system. And by doing that, actually, you will improve the health accordingly. Okay, so two types of prebiotic molecule normally you can find commercially. They are the galactose, oligosaccharide, or fructose, oligosaccharide-based polymer. So these are the two common ones. But beside on that, actually, on top of that, actually, there are a few more that can be used as a prebiotic as well. Right. So what are the challenges of LEB or the probiotic that I mentioned so far? I emphasize on the general statement or definition of probiotic. The live microorganism, the viability. So when you mention viability, it is quite challenging. It's a big challenge actually to us to secure or ensure they always viable, they always live or still living around. So we know that different culture, including us also, we have different ability to survive. Same thing also happened to different bacteria as well. So this imposes a big challenge actually for the probiotic product. And second point, different colonization, like I mentioned just now. Yeah, if let's say we have so many people around here, I put you into the new environment, can you mingle around easily? It depends on individual, right? So same thing happened to the new probiotic that you consume through your gut. And whether they can attach or not is a big question mark again here. Okay, the third point. Yeah, high maintenance cost because you want to ensure viability. It's not easy. So normally we have to keep into the certain cold temperature, normally at 4 degrees Celsius to have longer life period or shelf life period. And the following point is allergic properties that may due to probiotic as well. So it depends what kind of food you eat. They can produce also allergic like histamine. All right? And beside that, some report has shown that probiotic also have pro-inflammatory activity to those that have the inflamed gut especially. So it doesn't mean that when you take probiotic, it will give positive impact all the time. Right? Last, another one is high risk to immunocompromised patient like AIDS patient, ICU patient. Actually, probiotic can induce unnecessary side impact or fatal effect sometimes. Okay? And last one, still again, AMR acquisition gene through the horizontal transfer between the probiotic or between other genus within the gut system or environment as well. So this actually poses a huge impact nowadays, and that's the reason why the registration and the regulation body, they are very particular about AMR genes harbored in probiotic. Right, this slide shows that the threat of AMR issue. So it's been projected that, it's not a try to scare you off, but this is the report that given to us, that, about 10 million deaths per year will be attributed to the issue of antimicrobial resistance by year 2050. So it is higher than the cancer. So I don't know how true it is, let us see. But that is the alarming message that we should receive now. And what should we do? Right, so we have done a simple experiment. We try to isolate the Lactic bacteria from the feces of the chicken that we bought from the Sedang area. So the LDB they were isolated from the chicken feces. It shows that they have multiple resistance to the antibiotic that we use for treatment commonly. So it is quite scary. Right, so how about ICU patient? Okay, in year 2019, yeah, there's one report that reported by a very prestigious journal, yeah, which is come from Nature Medicine, and they reported that Lactobacillus rhamnosus, which is very, very famous of the probiotic that been available of sell in the commercial, and actually they cause bacteremia to the ICU patient. So what does it mean? They can find that they can isolate or they can detect, yeah, those Lactobacillus rhamnosus in the bloodstream of the ICU patient. And this one called the fatal effect to the ICU patient in US and Canada. Okay, how about paraprobiotics? Is it same like probiotic? No, it is different, slightly different, but still related to probiotic. So it is actually introduced by Travanti and Guglielmetti in 2011. And they try to define the paraprobiotic as non-viable microbial cells, either intact or broken or crude cell extract can be any preparation, but it means that they are not viable. They are dead. Okay? And when you consume this probiotic, they still give the same impact like the probiotic cell. Right? And here also the probiotic known as ghost probiotic because they're dead. They are not living. Yeah? Right. So actually the para means that you try to bypass is something like alongside with the preparation, and actually we try to go through we try to avoid the purification procedure for the para probiotic. In other words, they are not pure compound. They are mixture of myriad compound within the preparation. So another similar terminology is a postbiotic, which actually we work very heavily, extensively since year 2000. And what is postbiotic? It is soluble metabolic products that produce or derive from a defined fermentation process of live probiotic microorganism, which when administered in adequate amounts will confer beneficial effect to the host. And this definition and the postbiotic terminology was coined by Slingri and Racino in year 2012. However, we have done postbiotic from year 2000. And we have published the first article on metabolites on rats effect in year 2003. But however, we cannot publish easily during the time because nobody knows what is metabolites, what is postbiotic. And we have a lot of hard time to share our information that we generated from a research group. But we managed to push it to Pakistan Journal. And it has been recognized now, very well recognized now, the metabolites impact. Okay, so in year 2021, there's consensus actually try to propose by the International Scientific Association of Probiotics and Prebiotics and Probiotics and Prebiotics. And they publish in a very prestigious journal known as Nature Review Gastroenterology and Hepatology in year 2020 in May. And this consensus proposal has created a lot of debate after that. So, what have they proposed? They try to propose the probiotic become preparation of inaminate. All right, it's not common word in science. And microorganism and or their components that confers a health benefit on the host. Another terminology is definition is very broad and general. But it looks like it contains everything between the postbiotic and paraprobiotic. So what's going on? Or after that, what's happened? Okay, so there's two schools of thought, has been proposed or has been published from May 2021. All right, the origin of the proposal for postbiotic, they are not happy with the new consensus that proposed by ISAPP. So what happened? See, this is the first consensus that proposed by ISAPP, and later on, the initial proposal for the postbiotic and probiotic, they try to declare the proposal or the definition that proposed by ISAPP is not appropriate. So they say that, what they say? Too simple, fail to clarify the postbiotic. And of course, they try to fight back again. Okay, I think this is the first time that, to my knowledge, that the scientific world try to have a proper discussion through the article and consensus publication. So at the moment, I think postbiotic and parabiotic are not in the registration regulation yet. And I think we face a lot of problem to propose or to push our product to the market for the use. So I wish that FAO, WHO, or Kementerian Kesihatan Malaysia will look into that and try to see what should we do and what kind of definition we should adopt now to accommodate the new products that developed throughout the world nowadays, including Malaysia as well. Okay, so this slide summarizes the preparation of probiotic, everybody know, live cell. And if you try to lyse the cell, it becomes paraprobiotic, but without any purification. and it is, can say that, inactivated or non-viable preparation. But for postbiotic, we had to go through fermentation process by using a defined medium, depending on what you want to produce, what functionality you want to create. All right? And from here that you have to separate the viable cell and the fermented metabolite. You can, it is a postbiotic, and actually they contain various types of soluble metabolites. This is what we hold, this is what we propose, and this is what we publish until today. So, what we have done to verify the functionality of postbiotic and probiotic that we developed back in UPM since year 2000. So, we try to verify between the probiotic, paraprobiotic, and also postbiotic for their efficacy, and also try to derive or try to reduce their mechanisms. So this slide summarizes what we have done. So we try to isolate various culture or LB culture from the food sources from Malaysia, and we go through the fermentation process using the bioreactor. This 30-liter bioreactor is semi-scale or semi-pilot scale of the bioreactor, and we try to produce liquid one. After that, we use spray dryer to transform, become the powder form. And using these two preparations, we try to feed to various animal models, as shown here, which is the chicken, and this one for the egg, if correct, laying hen, for the egg production. And we also use rat. We started with the rats feeding trial, and also later on, we also fed to the piglets as well. And we also try to check or try to verify whether they have same trend of beneficial effect on the fish as well. We use tilapia fish as an example. Yeah? And later on, we also try to test on the lamb. Okay? And all of them actually show the same trend of beneficial impact. Right, so this slide summarizes all the beneficial impact that we obtain as what being reported for probiotic application or supplementation. Yeah, such as to prevent the intestine, inflammation, as well as anti-cancer impact, and immunomodulation, antioxidants, anti-inflammatory activity, and also reduction of serum cholesterol, very obvious for that one. And also we have antimicrobial activity. Right, okay. We have grouped the efficacy of functionality based on 3 categories. So very well spread efficacy based on the health promoting effect on rats and also livestock. The second group is known as a frequent efficacy. All right, and this one actually related to the in vitro and in vivo inhibitory activities against different pathogens. All right, and the last one is a bit unusual, which is a bit special. We call it rare efficacy, whereby they exhibit selective cytotoxicity activity on human cancer cells. Right, so in our postbiotic preparation with different strain, they also, we can identify and detected a few metabolites such as short chain fatty acid, extracellular enzymes such as proteolytic enzymes, cellulolytic enzymes, and hemicellular enzymes, and also exopolysaccharide, which produced by some of the strain. And of course, this is quite new to us because amino acid production industry normally carry out by Corynebacterium. But in our case, we managed to show that LAB can be a potential amino acid producer as well. And of course, they also produce bacteriocin because they contributed to the broad inhibitory activity. And diacetyl is another metabolite or secondary metabolite, you can say that contribute to the flavor, aroma, and texture of the food. All right, and this is a bit unusual also, yeah. For some of the LB, they can produce vitamin B as well and vitamin K. But in our case, we managed to show that they are the good producer of vitamin B9 or folate production. And deuterin is another molecule that can have inhibitory activity. Right, so this slide shows you the antimicrobial activities that normally that we do in the lab to determine the antimicrobial activity. So this is inhibition zone. If they have the inhibitory activity, the indicator bacteria cannot grow. So you have clear zone here. The bigger zone means that they are more stronger or potent, yeah, to that particular pathogens, right? Okay, so this slide summarizes the pathogen or food spoilage bacteria that we have been used to determine the inhibitory activity. And this group are the food spoilage bacteria. I think you all know very well, right? Bacillus subtilis and Salmonella, Staphylococcus, and Streptococcus, etc. And this group actually belongs to the same group of Lactic bacteria. But if they say they are the bacteria's producer, they actually can inhibit each other as well. And this is actually come from the aquaculture pathogens, such as Aeromonas and Streptococcus and Pseudomonas. And the last one actually is another culprit due to MR issue. And actually, Enterococcus actually is a LAB. But it becomes pathogen because they become vancomycin resistant. Vancomycin is an antibiotic being used for the treatment and also in livestock industry for some time. Right, so you can try to go through our article if you wish to know more about inhibitory activity. But I want to show you now why our postbiotic have a broad inhibitory activity. So most likely, we predict or propose that or hypothesize that actually due to the bacteriocin molecule that combine with the organic acid, combine with the deuterium molecule just now. So all of them work together synergistically to give a very broad inhibitory activity. Okay, so we have the bacteriocin structure. This is just a cartoonized structure. You have the cation domain, and you have the hydrophobic domain. So this general structure of the bacteriocin small molecule. All right, so what do they do? So when you have the bacteriocin molecule, they will try to hook up to the protein in the membrane. And together with another molecule of bacteriocin, they will combine such a way at the hydrophobic region. So what is next? This hydrophobic region will try to insert into the phospholipid membrane of any organism or microorganism. And by doing that way, you will create a pore. Or the cell wall will be broken up. And in this case, all the solute from the intracell can go to the extracellular component, or extracellular component also can enter the intracell. So if let's say you have organic acid outside the cell, you have the deuterium outside the cell, they can enter the cell. And by entering to the cytoplasm, the organic acid will be dissociated to become proton and also to become conjugate base. And others also can be happen. And in this case, when you have the proton being dissociated from the organic acid, for example, the lactic acid, acetic acid, so it will create a low pH in the cytoplasm. So in order to survive, the cell will try to remove the proton from the intracellular. And this one involves the proton pump. That means the bacteria will keep pumping out the proton, because after that, it pump out, it will come in again, right? Because it's already broken the cell wall. So sooner or later, the cell will be exhausted and they will die off because of that. Right, so this table shows you the organic acid that can be produced by our 6-strain culture here. So they produce lactic acid, acetic acid, and formic acid in different concentration. Right, so what is the benefit of organic acid? Besides become inhibitory activity or give the inhibitory activity, actually they also can stimulate the mucus production. Mucin production actually is very important to safeguard our gut health. All right, and at the same time also they can inhibit or inhibition of the inflammation activity, yeah. And also they can have the IgA production, and also actually they can promote the regulatory T cell. Actually, this is a very important immunoglobulin cell, or not immunoglobulin cell, but they are the immune cells that involve into the safeguard of our immune system. Yeah, by having that, actually it can protect from various pathogen and invader into our body. And at the same time, also organisms being reported can repair tissue as well, especially the gut tissue. Right, so what are bacteriocin? Actually produced by certain algae. Not all algae produce bacteriocin. Okay, if you have the algae can produce bacteriocin, it's something like a lucky draw. Or it can be like it's extra features of the lactic acid bacteria. And actually, it is a group of the ribosoma synthesized antimicrobial peptides of protein. And it is a very small molecule. It's between 30 to 60 amino acid residue. And actually, this molecule can associate with carbohydrate, lipid to become larger molecule. All right, so altogether, actually got 3 classes of bacteriocin that reported so far. And basically, they have the all inhibitory pattern to certain bacteria and also foodborne pathogen. And those produced bacteriocin, they have the resistance to their own bacteriocin. So it means that it will not be killed by its own bacteriocin production. So we have tried to check why our postbiotic have so huge or broad inhibitory activity. So we try to check what kind of bacteriocin harbor in our culture. So we check various genetic determination, such as structure genes, modification genes, regulatory genes, and also export genes, immunity genes to the producer cell. So with the help of Prof again, so we managed to try to amplify 2 bacteriocin genes from the culture that we work closely. All right, so here it shows that 2 bacteriocin genes actually harbor in our culture, which are the plant EF and plantaricin W or plantaricin EF, which is a class 1 and class 2 bacteriocin. Means that our culture harbor 2 classes of bacteriocin here. So that's the reason why it has very broad inhibitory activity. And we also try to check their structure, whether they are the new DNA structure or genomic structure or not. Yes, they are different or compared to other reported culture here. Right, so after we have the bacteriocin, what should we do? And this is always involved in the bioprocess technology. Right, so in order to make product viable for the industry, we have to do optimization because we have to reduce the cost and increase the yield at the same time if possible. So this is what we have done. All right, so we have refined the medium using the statistic software run as a response surface methodology, and we can try to optimize or try to study the interaction between the elements or the growth medium that involved in the production. So for example here, right? So if this central point is the optimal level that we should adopt for each of the interaction between the growth medium parameters. So in this case, by having the refined medium, as shown here, we can reduce or we can increase the inhibitority to 108%. At the same time, we can reduce the cost to 85%. So deuterins, another inhibitory compound, it has a bound function compound with the hydroxy group and also the aldehyde group. So it is also a very potent inhibitory compound. Right, for folic acid. Why folic acid? Why we emphasize on folic acid so much? Because actually it is a very important ingredient for our chromosome synthesis. They involve in the base pairs of chromosome. Without that, the cell cannot grow. None of us can grow easily. Okay, for repair purposes of the cell damage as well. So for another function of folate is prevent colon cancer and growth retardation and also anemia prevention. So the last one, actually, this is very important for those pregnant mother or lady. If you want to ensure your baby grow well, you should have the folate supplementation, because this one can prevent the neural tubes, defects in the newborn. Right, so actually we try to compare our culture, UL4,
compared with those being used in the commercial for the production of vitamin B9. It shows that our culture are more superior than the commercial strain that used for the production. So next one, I want to change your paradigm to another development that we have developed. We try to use the solid state fermentation, a bit unique of a fermentation that you know of without much of the water, to develop various compounds or can be used for the feed industry or livestock industry. So we make use of several agro waste. Yeah, for example, here with pollard, pumpkin cake, cocoa pod husk, and a mushroom waste, and also soybean meal. We try to develop something valuable, some valuable product from there through the solid state fermentation. And I think this news is a bit old. Yeah, it's been reported in year 2011, but the same history come back nowadays. It's a food safety, food security problem again that we are facing very, very badly at the moment throughout the world, including Malaysia. So feed costs again, right? Into the impact on the poultry industry. So what should we do? Actually, we were asked by that time to make use of lactic bacteria to develop something good from the local resources as an alternative, yeah, for the livestock industry. So what have we done? We try to make use of our culture and try to go through the fermentation process. And for your information, all right, PKC has been used for ruminant, right? And they can degrade the PKC very well, but not for monogastric animal like the chicken. So what should we do? We have to process further before the monogastric animal can consume the PKC well. Okay, for your information again, actually PKC contain quite a lot of nutritive compound. For example here, they contain 53% galactomannan, which is prebiotic actually. And at the same time also, this percentage of protein can be taped on to further degrade it to become protein source a little bit, but not much, but still there, okay? And another one actually is the cellulosic material. So actually we are targeting to 3 components there to further develop them to become a useful product. So in order for the cellulosic material, actually these are the cellulose compound, hemicellulose polymer. They are quite complicated. And another one is cellulose is much more simple, which is a polymer of beta-1,4 link glucose unit. Okay, in order to degrade cellulosic material, we need at least 2 groups of enzymes known as cellulases and hemicellulases to work synergistically. Right, so these are the enzymes that are being grouped into the cellulases, and these are enzymes that group in the hemicellulatic enzymes. So you need all of them to degrade, yeah, the PKC or cellulosic material efficiently. Okay, why we use solid state fermentation in the product developed for livestock industry? Because actually we can harvest the product much easier without much of the downstream processing process. And actually what, according to Pendy 2010, it is a process that involves the growth of microorganism on solid materials in the absence or nearly absence of water, yeah, free water. Means that they still have moisture, but they don't have free water dripping or moving around. Not like the suspensions types of the fermentation process. Right, so this slide summarizes the advantages of solid state fermentation. So here you should see that they are simple process, low processing cost, low energy consumption compared to the normal bioreactor system, and low risk of contamination, simple design machinery, and less effluent. Release means that you can have less pollution to the environment, and it is also have a high product yields and resemble the nature habitat of the microorganism that you use for process, and it is easier downstream processing because not much water to remove. So basically, solid state fermentation normally being reported for fungi, not for bacteria. Very rare that bacteria can carry out, yeah, solid state fermentation condition. And if you can use bacteria, it's better and more beneficial because you can avoid the mycotoxin production due to the fungi growth. And it is safer to be conducted by bacterial species. So in our case, we try to check the extracellular enzymes activity. It shows here that we have various protein activity that which is very, very versatile because they're active from pH 5 to pH 8. And this culture, actually, they are superior. Yeah, proteolytic enzyme activity here. I tried to cut it short because it's too long. Okay, in this slide shows that we also can obtain various types of cellulitic and hemicytic enzymes activity at the same time. So it means that they are versatile extracellular enzyme producer. Okay, and we compare, try to rate the culture, which one is better, because we need to select for our solid state fermentation of PKC. And we select 3 of them as our angel here. And looks like, surprising to us when we try to have that activity and result, it shows that the culture we select, Ybhg11, Ybhg14, R11, actually they can grow quite well, up to 14 days under the nearly 0 water condition on PKC. Right? So it means that they are versatile. They can grow. They can maintain the growth, even though the growth of the population is higher than the initial population here. So what else? We check the enzyme activity also after the fermentation process, and it shows the same profile that we got before, but of course slightly different. And actually those extracellular enzyme activity work synergistically and concerted manner. Okay, you can see that actually this enzyme produce more at the beginning, but this enzyme produced at the center of the incubation period. This also same thing, but this one actually is throughout. So all of them actually they work in the concerted manner to degrade the cellulose and also amylose effectively. Right, so you see that it's very, very interesting. You see that they are hanging around with some appendage, and our quick structure, we call it exopolysaccharide. So means that you see that they all hang to the cell wall, they can survive very well. At the same time, they also release the extracellular enzymes to degrade the polymer accordingly. Okay, right, so after fermentation, we check the quality of the PKC, fermented PKC with the proximate analysis. It shows that here, they can increase the protein content at the day 6 of incubation, and also they can reduce the cellulose content at the day 12, and they also reduce the hemicellulose content at the day 4. All right? So it means that actually these hemicellulose have been changed to become prebiotic now. Okay, how about mineral? The mineral, they reduce copper very substantially, very significantly, while increasing the good mineral that we want, such as calcium, iron, magnesium, manganese, potassium, and zinc. Right. So for the application of amino acid, I think we know that amino acid is a building block for protein. And nowadays, amino acid being used for many, many applications. For example, for livestock, nowadays they use lysine, threonine, tryptophan to improve the growth of livestock. At the same time, also actually they contribute the largest share of the amino acid market. At the same time, also for food industry, MSG has been used, and aspartame to be used as a replacer for the sugar for the food production. And for the pharmaceutical industry, tryptophan has been used as a drug now, right? And for the cosmetic, also some of them being used as the moisturizer. So in our case, what should we do? What we have done, actually we have checked the ability of various culture to produce amino acid. You can see here, actually all of them, they produce different types of amino acid in the metabolites. At the same time also here, right, another study here, also we check the lysine production. Not many of them can produce lysine, yeah? And proline actually is a good amino acid produce collagen. Okay, it's been used a lot in the cosmetic preparation nowadays, right? Okay, so this is the culture that can produce 15 types. Okay, and you can see here, these two cultures can produce lysine, but not all of them. Right, so what we have done, we further optimize it to reduce the cost, to increase the yield. So here it shows that we can improve the 2 times threonine production. At the same time, we reduce the cost to 8.25%. How about the tryptophan? Same thing again. We can improve 157% of tryptophan production and reduce the cost of to 11%. Sorry, 11, yeah. Okay, so conclusion, can lattice bacteria, are the lattice bacteria be the biotransformation for the sick biomass? Yes, all right? And for the gut health, okay, is another important criteria for us. So we try to check our postbiotic can improve the health or not. As we know that gut harbor trillions of bacteria, harbor the trillions of bacteria, fungi, and also microbes. And they can be generally grouped into 6 common or major phylums of bacteria, such as Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. These are the 4 common ones. Remember just now I said that Lactobacilli belong to these Firmicutes, and Bifidobacterium belongs to this Actinobacteria phylum. So if you have these 2 in the gut, you can avoid a lot of dysfunction. And basically, the gut also involved, the gut microbiome involved in our digestion, immunity, and also the metabolism pathway, and also our mood as well. Some of you may feel that, oh, today is not good mood. Today I'm so stressed. So what should we do? To ensure that you have a good gut health, because actually, this one actually related to the neurotransmitter produced in our gut, such as GABA, all right, dopamine, and serotonin. Actually, all of them derive from amino acid. Okay? So the rest, I think, they also produce vitamin, we know already, right? Okay, so what is GABA? It's a neurotransmitter. Actually, it's an inhibitor of the neurotransfer. And this one, actually, you can try to have more relaxation and calm mood if you have GABA. Okay, actually GABA has been subscribed as a relaxation for the pain and for the stress as well. Okay, how about dopamine and serotonin? They're known as happy hormones. You have both means that you are happy throughout the day. Okay, so they are the essential neurotransmitters to have in our gut system. Right, for those dysfunction gut, you will have the irritable, bowel disease syndrome, and some of you may have allergic and eczema and asthma impact as well. So nowadays, I think a lot of children, they have eczema and allergy effect. Maybe due to the gut microbiome is not balanced. Right, okay, we try to check, those compounds. Our postbiotic can emulate or modulate our gut health or not. Okay, so our rat trial shows that they have a good impact on the growth rate and also, but not the feed conversion ratio, they are no different. But they have an impact on the fecal LAB population, which is good population, and they suppress the enteroparasitic count like E. coli, Salmonella bad guy. And they also reduce the pH very significantly. Okay, so this slide shows the impact on the chicken trials under the heat stress condition. And it shows that all of them, they improve the villa heights, basically, but actually for the jejunum region, and also, but not for the crib death, but they also have the, those feed with the postbiotic, they have better villa height compared to the control here. Okay, for the antioxidant activity, for the microbial population here, so we have a better population of LB, which is Lactobacilli bifidum, but we have the bad guy, we reduce the bad guy here, and they increase also the, decrease also the pH of the gut of chicken. So what should we do lately? What I've done lately, we compare the parabiotic and postbiotic feeding to the chicken. And it shows that both preparation of probiotic and postbiotic, they modulate the gut microbiome to be more firmicate and bacteroides system compared to the control. How about blood cholesterol reduction? Can they do the same magic like probiotic? Yes, they can, because you see, feeding with a spray-dried postbiotic, they reduce the cholesterol as compared to control. We also try to compare the assimilation of cholesterol in vitro by the strains that we work on. Basically, they're different assimilation of a cholesterol profile. And we compare now with the feeding of the probiotic. This is a profile of reduction of the cholesterol. How about postbiotic? They do the same magic as well, but slightly different level. Yeah? So it looks like all of them reduce the cholesterol, same trend like the probiotic. And we try to check also the antioxidant activity. Sorry, it's not the cholesterol profile of the blood that of the chicken that fed with the postbiotic. It looks like they improve, the cholesterol level, TAG level, which is the bad guy of this one, and they increase the good cholesterol, HDL and VLDL, etc. Okay, how about antioxidant activity? Basically, they improve all the antioxidant activity in the blood system as well. Okay, how about immunomodulation impact? As we know that we have 5 classes of immunoglobulin from IgA, IgG, IgM, IgE, and IgD. IgA is the immunoglobulin that normally you can find in our gut system, and IgG is general immunoglobulin, that which is second largest, or second dominance of immunoglobulin, and this is the first immunoglobulin that will be produced when you have the invader, IgM. IgE is the immunoglobulin that is responsible for the allergic. And this one, IgD, is a bit new, and not much information has been available until today. And in our trial, it shows that the postbiotic, RI11, is a good inducer for IgG, IgA, and IgM, compared to the other 2, preparation and control here. Okay, you can read up more if you want to know more what our trials for the chicken. And for this approach, or for this observation, or the result we obtained, we filed the patent to 7 countries, and those highlighted in yellow, we have already obtained the patent, officially. And we also filed a patent for the aquaculture, application, and these are the countries that have already granted the patent. And now we try to target AGP issue. So we hope that we can make use of that postbiotic also to replace the antibiotic as a growth promoter. You can see here, compare here, this is the same bacteria, and you see that inhibitory become very, very alarming here. Right, so this is the spin-off company from UPM, which is known as Ecobionic Sembilan Berhad. We try to deal with the postbiome for the feeding trial and also the fermented PKC here. Okay, how about cancer treatment? Anti-cancer effect. We try to make use of the postbiotic as an adjunctive cancer treatment as well. So you can see here, this is a distribution of cancer incidence throughout the world in year 2020. I cannot find 2021. That's only the latest information I can find. So it shows here that, here it's about 19.3 million cases that, and 10 million cancers deaths in 2020. So actually, it's keep increasing every year, every day as well. So what should we do? And here, it shows that this is a dominant cancer incidence of male. We should start from lung, and prostate cancer for the organ, and colorectal. And for the woman, it's always the breast cancer, right? So that's the reason why we try to see our postbiotic have anti-cancer impact on various cancer cells or not. This one based on the in vitro activity. And in year 2016, one of my students, Tang Hui Keng, she tried to propose a new terminology known as cytobiotic. So what is cytobiotic? It is a postbiotic that exhibits cytotoxicity effect on human cancer cells through the apoptotic mechanism and can be developed as a natural chemopreventive agent for cancer, for overall, help maintenance, cancer prevention, and also as adjunctive therapy for cancer treatment is quite lengthy. But there's a few things that we want to include here. Apoptotic and chemopreventive agent, right? So what we have done, we try to check the toxicity, whether our postbiotic toxic or not. So we use various types of, blood cell, red blood cell, and it shows that they are pretty safe compared to the control of water and sodium. SAS is sodium disulfate, which is chemical that break the cell. And we check with various cancer cells here, and it shows that they have different cytotoxicity activity against the breast cancer cell, you know, and different type of cancer cell. And here, they're actually very potent to the leukemic cell. Right, so in order to kill a cell, normally two mechanisms involved, which is the necrosis and apoptosis. Apoptosis is the one that's favorable for the cancer cell killing. And because this one will not cause inflammation, this one will cause inflammation upon rupture of the cell, right? And our treatment with the postbiotic, it shows that all of them, the cancer cell actually go through the apoptotic mechanism. And this control, nothing happened. Okay, and we also verified with the flow cytometry analysis, it shows that this is the control. All the cells actually remain healthy up to 72 hours, but our treated cell actually, they have entered the late apoptotic phase. Okay, especially the 72 hours, they all go to this direction. So it means that they are very specific, right? Okay, so we have found the patent as well, and here we have up to quite a number of countries that granted the patent to us. And this patent has acquired by My Miracle Ybhg in early this year, January. All right, actually they also acquired the patent of nutraceutical patent as well. And this is a product that will be marketed by the company. You can find some product outside as well. And we also check on the E. coli metabolite. I think I quickly go through. Actually, they also have the cytotoxicity and also anti-inflammation and activity, and also the anti-inflammation activity, and also for the anti-inflammation genes as well. Right. So, okay, general conclusion. Probiotic health impact depends on lifestyle or their metabolites. I think you all can answer, right? Okay. Okay, so postbiotic is strain specific. Each strain produce different combination or myriad compound of postbiotic. So all of us have to characterize it carefully, detailedly, if you want to use as a product development. And normally our postbiotic here, they contain various types of enzymes, biotic compounds, and hydrolytic enzymes. We also have the antimicrobial activity, antioxidant activity, anti-cancer activity, anti-inflammation activity, and immunomodulation activity. All right? And generally, they improve the growth and gut health. Yeah, no doubt. And this slide try to highlight the futuristic probiotic actually derived from paraprobiotic, postbiotic, and cytobiotic because they do not involve the lysol. In terms of preparation and product quality, they are much more safer and can last longer. Right? Okay, so let food be the medicine and medicine be the food. All right? Okay, so almost last, huh? Okay, last question. Can we use Lactic bacteria as a promising alternative to achieve the sustainable development goal? How many if year? Is that all? This I can tick off, almost all. But how? Maybe I would like to invite you to join our 12th conference of Asia Federation of Lactic Bacteria, which will be held in Kuching 10 to 13. So from there, you can get more information. You can go to the website and register. It's open for registration now. And you can have 7 themes of conference that will be discussed in the conference. And we have more than 20 international speakers from Asia country, also from Europe as well. So please join us. This is the first time that we have an international conference for lactic acid bacteria in Malaysia. Right, so to conclude everything, these are the paradigm shifts that we achieved in our research from year 2000 until now. So these are the, to improve the inhibitor activity and to make use of the extracellular enzyme for the biotransformation process, and also to make use to become the human health supplements, feed additive, and also we try to use it for vitamin B production, amino acid production, etc. So I would like to thank UPM, before I end my speech. I'm a bit long, and I would like to thank UPM to include me as one of the member in this big family. And I want to thank Pro-Chancellor, Accommodation Professor Emeritus, who really introduced me to research work in year 1990, actually. I worked as a research assistant in the long holiday semester. After the first year of the long-term holiday, I worked there and have been trained up as a researcher until today. And I also would like to thank Professor Dr. Gulam, the former Dean of Food Science Faculty. He is the one who introduced me to the world of lactic acid bacteria. And he convinced me and persuaded me and tried to ask me to help in the research activity of lactic acid bacteria. And this is how I transformed myself from the plant-based research, work on the 2 enzymes that I isolated from the glucosinolate, brassica crop, and now I work on the microorganism again on lactic acid bacteria. Right. Okay. So I also would like to thank Y College. This is Dr. John Rossiter. We visited Y College, I think sometimes back 6 or 5 years ago, and but Y College no longer there. Okay. It has been acquired by Imperial College. It's known as Imperial College at Y. Right, but it is a very beautiful village and very beautiful college. I really miss it very much. Okay. Right, I also would like to thank Ministry of Higher Education, Ministry of Science, Technology and Innovation, Ministry of Agriculture and Agro-Based Industry, and also Malaysian Technology Development, Corporation Sdn Bhd. You are the one that support me throughout my research, life here. Right, and of course, industry partners like Malaysian Flamir, Mr. Tay, Engineer Azrari, and Mr. Oya. Thank you for your support all the while, and My Miracle as well, and others, company who has helped in certain activity. Right, so of course, I would like to thank my big sister in UPM, who is a Professor Dato' Prof. Raha Abdullahim. Actually, he always extends his hand when I need it. Okay? This is Prof. Raha. And this is Prof. Rose, right? You're not mistaken? Yeah? And this is the photo that taken during the visit to the layer farm. And this is the first time that Prof. Rose introduced us to layer farm because we need to develop the bioactive molecule and also the fermentation process to develop the feed ingredient. Right, and this is the house of Raha that her mom can cook very well, Lasa Johor. Okay, and we also here feed with the Lasa Johor. Okay, and again, thank you, for the both hands that are given by Prof. Raha and also Prof. Rose. Prof. Rose actually is a fermentation technologist. And she helped me in the development of the bioprocess technology as well and fermentation activity. Okay, this is the photo that we taken during the Sabah trips that we isolate the LB from Bambangan. Okay, so this is the fruits that actually only can find indigenous fruit from. Okay, so we prepare the fermented fruit in a cottage, household by this, auntie, and she teaches how to do that, and we bring back the sample for isolation purposes. Right, and yeah, this is the, you see, who is this guy? Do you know him? Our Patla, right? Our former PM. Actually, he drank our postbiotic before. In year 2006, at the AAPI exhibition, this is the first PM's, exhibition award. And there's a joke that he got the second merit after that. Right, so this is the APC RMC award that I received in year 2006, yeah, due to the first commercialization activity that I managed to achieve, right? And okay, I would like to thank my committee member, used to be the committee member of the publicity and also the promotion. Yeah, I've been head for 2 times for this committee for the past 20 years, and these are the activities that we have gone through. Thank you for the committee that we enjoy a lot actually during the exhibition and publicity activity. Right, and this is my good friend, the alumni of UPM. Yeah, I think some of you are here, right? Yeah, we had the gathering one day. And this is a new group member that in IBS that I joined now, ChemRes Laboratory. Okay, of course, I would like to thank my postgraduate and graduate students who really work hard to fulfill my vision and mission of research on lactic acid bacteria. And I think you have worked very hard until we can achieve so much of, until today. Okay? And this is some of the exhibition of the research. Yeah, I think Dr. Yinsi, do you still know him? She is my first master student PhD, and who really started the exploration of the LB that we have the culture to work with until today. Yeah? All right? So, yeah, I would like to give credit to her, actually. All right? And, of course, these are the students. Dr. Anu actually worked on the aquaculture. And now Dr. Chua Leon actually worked on the anti-cancer impact. All right? And this is the gathering that we had before. And this is the latest PhD student, yeah, Acharya from the Thailand, who worked on the E. coli metabolites. Yeah? So we counter-proof that now. Doesn't matter what species you produce, as long as they have produced, useful metabolite, actually they have health impact as well. But provided we have to ensure that the culture is not pathogenic. Okay? Okay, so this is another exhibition that we involve with the presence of the Selangor Sultans. Yeah? All right. Yeah, this is per chapter, exhibition that we won the award for the exhibition as well. And of course, yeah, I would like to thank also Malaysian Society for Microbiology, which I involved since year 2000. And I've learned a lot actually. And I also, have a lot of experience with the society management and activity. Yeah. And of course, Asian Federation of the Lactic Acid Bacteria, which I have to involve pretty much now. Okay, last but not least, okay, I would like to thank, yeah, this group of people, yeah, for the inaugural group preparation. Without the help and from UPM Press, I don't think we can produce the book on time. Okay, and I think they have designed very nice cover page for the inaugural book. Right? Of course, I would like to thank the committee who has worked very hard for the past few months to arrange the inaugural of today. Thank you very much, everyone. Okay, lastly, all right. Do you know who this guy? Sometimes I confuse who is this guy. Because he plays so many roles, all right? But he always the hero of the house. Okay, thank you very much. And with this support, I can move on until today. Yeah? Without the support, I don't think I can make a miracle here. Right? And, okay. Right, but unfortunately, my eldest daughter cannot with me. Yeah, she's in Australia at the moment due to the exams, but I hope that she's living well there. Okay, and actually I would like to dedicate my inaugural to my late mother, Kwek Susim, and also my late grandmother, Ybhg. They are the one who raised me up without any fear and without any, do not give up even though how hard is the situation. All right, so with that, I would like to end my lecture with a quote of the day, okay? If you do something new or innovative, expect trouble, all right? But think critically about it, because if you are wrong, you want to be the first one to know. So this is always innovation. Yeah, about involve innovation. So for the young scientists or young students, new students, please bear in mind, don't afraid of new. Okay, and this is the advice actually given by the astrophysicist Eugene Parker, that given to the early career of the scientists back to NASA there. And actually he passed away at age 94. Right. Oops. That's all. Thank you very much for your attention, and to be patient with my long inaugural lecture.
Terima kasih yang berbahagia Profesor Dr. Foo Hooi Ling di atas penyampaian syarahan inaugural beliau sebentar tadi. Seterusnya, majlis dengan ini mempersilakan yang berbahagia Profesor Dr. Insinyur BT Hang Tuah Baharudin, Timbalan Naib Chancellor Universiti Putra Malaysia, ke atas pentas bagi sesi rumusan syarahan inaugural pada pagi ini. Majlis dengan segala hormatnya mempersilakan.
Terima kasih saudara pengacara majlis. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi taala wabarakatuh, dan salam sejahtera, selamat pagi kepada semua. Pertamanya, yang berbahagia Accumulation Emeritus Profesor Tan Sri Datuk Dr. Syed Jalaluddin Syed Salim, Pro Chancellor Universiti Putra Malaysia. Yang berbahagia Profesor Dr. Suhaimi, Dekan Fakulti Bioteknologi dan Biosains. Tentu sekali yang kita raikan pada pagi ini, iaitu Profesor Dr. Foo Hooi Ling. Terima kasih atas lecture yang amat menarik. Saya akan kongsi wrap up sedikit selepas ni. But I'm from the engineering background, so bear with me kalau benda tak betul lah. Tolong betulkan. Saya juga ingin ucapkan terima kasih kepada Datin Paduka Prof Khatijah, ada dari belakang. Dia menyorok kat tepi tu. Instrumental to Prof Prof Foo punya pasukan. Tadi saya jumpa Datuk Paduka Prof Raha, tapi saya tak nampak dalamnya. Ada tak? Oh, dia ada meeting lain kot, saya rasa. Pegawai-pegawai kanan universiti, pengurusan Fakulti Bioteknologi, dan juga rakan-rakan akademik yang lain, rakan-rakan industri yang hadir pada pagi ini, semua tetamu-tetamu Prof Foo dan rakan-rakan strategik beliau, dan juga para pelajar yang ada pada pagi ini. Prof Foo sediakan some basic points, key points. Saya tak perlu go through the key points because I'm trying to remember certain things. One of the things yang saya rasa very interesting is that over the span of 23 years and still going on strong, it is amazing that we can hear such a very interesting talk. And then all the development throughout your career has been instrumental in a lot of things. And then we know that the potential is huge after this, huge. Just to recall, the Vice Chancellor mentioned earlier, Prof. Foo's involved in 33 patent filings altogether, either by herself or with her colleagues. There are 33 altogether, and out of that, so far, 22 is yours on your own, in your own research, and then 5 has been commercialized. Ada 5 yang dah dikomersialkan, dan rakan industri itu ada di sini. Some of them are here, was part of the team yang believe in the research output. Kalau saya tahu awal, mungkin the way saya negotiate dengan industri probably different lah, Prof. But the next 17 tu yang ada lagi in the pipeline tu, come to my office, we can discuss better return for you as well as for the university lah. But so a lot of things we believe, then kita percaya as a university we would like to encourage our staff to pursue this idea, to pursue this, be innovative. Yang itu yang kita galakkan. And then not only be innovative in your research area, but making an impact. Ada impact yang kita rasakan perlu. So this has been clearly shown by Prof. Fu's work and her team, and then we would like to see these things, carry on further lah. I like the GABA just now, dopamine and serotonin, kan? If you can isolate them and then produce that for me, then I can have happy mood all year round, Prof. Yeah. Postbiotic, right? Okay. Yeah. So I think everybody in this hall understood probiotic, prebiotic, postbiotic. Hari ini saya belajar banyak thing. And then parabiotic, ini paratroopers lah basically. Parabiotics. And then cytobiotic, that's the new one. Which is I think work closely now with Datin Khatijah punya team on cancer research, I hope. So these are new things in the pipeline, and we hope that continuous support from the industry will be good. And we know that some of these are very big in terms of value for commercialization. One of the things yang kita buat di university is we are opening up our relationship with industries, not only local, because sometimes some of this product may not be readily acceptable here dalam negara because the market is small. So our office is actually in discussion with international partners, because product to market, bigger markets are actually in Europe, in America and things like this. So we are hoping that some of this product can be introduced at a different platform, so that it can be ready to the market much nearer and much bigger market. So then if it's come back here, people trust it more. Kalau Prof. Fu punya produk, we as a academician we understood it is good. Tapi kalau produk tu brand Malaysia, not many believe in the product. However, if the product is transferred, say to Yakult, and then Yakult bring it back, semua beli. So those are the strategy moving forward lah, some of the office that we're trying to do. I'm not going to be long, it's not my inaugural lecture. Saya punya tak tahu bila, lambat lagi ya. Enzyme is one of the things, I think, which is a good thing. You've done a great job into that. One of the things yang recently that I'm engaged with a team from Japan and from Europe, they're also finding out, of course this is not lactic acid lah, but other acid, they're looking at enzyme that will dissolve and plastics, so that it can be reused, recycled, in a sense that it goes back into its original form. It can be, but the concept is like lactic acid bacteria lah, lactic acid bacteria concept lah, but it's through enzyme process. So those are also a big research, and then those who are in bioscience and all, there are huge funding for enzyme, looking at natural enzyme that can dissolve plastics to go back into its natural form. It can be reuse and reuse and reuse, so on and so forth. So I'm not going to be too long anymore. So again, I would like to thank again Professor Foo for her inaugural lecture and her great achievement over the years in developing new products. I know that she found love in the career in university, and also she found her love in her life also here, saya rasa. Kan? Prof, if Prof Loh is hero, Prof Loh is also my hero in my office. So it's a pengarah UCTC. So, and a great couple. We are hoping that they contribute a lot to the university. And then, can we all give another big applause to Prof Foo? Jadi dengan itu saya sekali lagi mengucapkan terima kasih atas syarahan inaugural Prof. Terima kasih. Thank you.
Majlis merakamkan setinggi-tinggi penghargaan kepada Yang Berbahagia Timbanet Chancellor atas kesudian beliau merumuskan syarahan inaugural pada pagi yang barakah ini. Yang Berbahagia Pro-Chancellor dan sidang hadirin sekalian, saya mohon menyampaikan acara seterusnya dalam bahasa Inggeris. Ladies and gentlemen, Professor Dr. Foo Hooi Ling has commanded an outstanding excellence and prominence in scientific research. Transcendent of these accolades, today we are honored to witness the exchange of patent acquisition document between UPM and My Miracle Sdn Bhd. This exchange involves the acquisition of 2 patents, of which Yang Berbahagia Prof. Foo is an inventor. The first one being tumor cytotoxic agent and methods thereof, and the second, probiotic composition for nutraceutical products. Ladies and gentlemen, for this purpose, please enjoy the video montage deliberating these innovations prepared for us today. Ladies and gentlemen, it is now my honor to invite Yang Berbahagia Deputy Vice Chancellor, together with Yang Berbahagia Professor Shuhaimi Mustafa and Professor Dr. Foo Hooi Ling, onto the stage. Ladies and gentlemen, I would like to invite Mr. Chin Kok Tian, Managing Director of My Miracle Sdn Bhd, to exchange documents for patent acquisition with Yang Berbahagia Professor Dr. Insinyur B.T. Hang Tuah Baharudin, Deputy Vice Chancellor of UPM. Thank you, Mr. Chim Kok Tian. I would like to invite Yang Berbahagia Deputy Vice Chancellor and Professor Shuhaimi to remain on stage for a photo session with Professor Dr. Foo Hooi Ling. Ladies and gentlemen, behind every successful person is obviously a loving and supportive family. It is my pleasure to now invite the family of Professor Dr. Foo Hooi Ling onto the stage with our Div Kormat for a photo session. Prof. Lo, we may want you to remove your mask, although we all know you. It would be great to see a smile. Thank you, Prof. Lo and family. I would like to ask for Prof. Foo, Yang Berbahagia Deputy Vice Chancellor and Prof. Syami to remain on stage. I would like to now invite the top management of the faculty onto the stage for a final photo session. Thank you, top management. Thank you, Yang Berbahagia Deputy Vice Chancellor and Prof. Shuhaimi. I would like to ask for Prof. Foo to remain on stage by herself, if it's possible. Ladies and gentlemen, as we all know, Prof. Foo wears many hats, both inside the academia and outside the academia. For this purpose, I would like to invite a representative from the Malaysian Society for Lactic Acid Bacteria to present Prof. Foo with an honorary token of appreciation in commemoration of her inaugural lecture today. Thank you, Dr. Adin, and once again, thank you, Prof. Foo. Hadirin sekalian, bunga dedap di atas para, anak dusun pasang pelita. Kalau tersilap tutur bicara, jemari disusun, maaf dipinta. Dengan berakhirnya majlis sebentar tadi, maka Majlis Syarahan Inaugural Prof. Dr. Foo Hooi Ling pada pagi ini telah sampai ke penghujungnya. Majlis ingin merakamkan ucapan terima kasih di atas kehadiran pada hadirin sekalian. Bagi memperingati majlis inaugural pada hari ini, majlis ingin memohon kerjasama para hadirin sekalian untuk turut sama yang berbahagia Tan Sri Datuk Pro Chancellor dan juga Timbalan Chancellor dalam sesi fotografi pada pagi ini. Bagi tujuan ini, saya mohon jasa baik tuan-tuan dan puan-puan semua untuk bergerak ke bahagian kanan Dewan. Tuan-tuan dan puan-puan, jika boleh kita boleh bergerak ke bahagian kanan Dewan bersama dengan Prof dan div-div kehormat kita.
Tidak luntur, semangat membara, segalanya untuk Siti tercinta. Seruan al-falah terus bergema, imam matlamat teras utama, menara ilmu diperkasa, berilmu berbakti untuk selamanya. Ke bukit bersama didaki, kelurah sama dituruni, ilmu diteruskan bersama dikongsi, Uni Putra terus terbang tinggi. Seruan al-falah terus bergema, imam matlamat teras utama, menara ilmu diperkasa.
Baiklah, jika semua sudah bersedia, kita boleh mengambil fotografi.